Good afternoon, everyone. Let's sit down and I'll introduce Michael Van Nuland, who is the next candidate in our plant ecology search. So thank you all for coming this afternoon. Michael is currently a postdoctoral research scholar at Stanford University, and he did his PhD at the University of Tennessee, and he's interested in plant microbial interactions, and I will let him fill us in on the details. So let's welcome our speaker.
Yeah, thank you very much, and I appreciate all of you coming today and really giving me the chance to share some of my research that I'm really excited about, which, as Michael mentioned, is going to be focused on plant-microbe interactions, how they influence species ranges, and the structure of terrestrial ecosystems. And before I get started, I want to quickly thank the huge amount of support that I've had on these projects over the years. All of the work you're about to see has been the labor of love from an enormous and helpful amount of people, from undergrads helping me repot thousands of plants in greenhouses to sequencing facility operators and mentors and a diverse range of funding agencies. So none of this would have been possible without the support of these people and these agencies. So one of the most foundational questions in plant ecology is, where do plants grow and why do they grow there? And people have been asking and trying to answer this question for centuries, right? If you think about Humboldt's early illustrations of plant species ranges across mountains in South America, or warming beginning to link plant morphology and anatomy to the different types of habitats that they occur in. And these questions are still important today for modern ecologists, because the factors that drive species distributions, or the things that affect range limits, ultimately tell you something interesting and important about the ecological and evolutionary forces acting on those species. And this becomes even more critical if we start to consider how ecosystems are changing globally. And while we've obviously made tremendous work and made tremendous strides asking and trying to address this question, thanks in large part to the explosion of things like functional trait ecology, or just our general ability to link climates and plant distributions and competitive interactions to think about how species are positioned on the landscape, I would argue that there's really been one major component that's been left out of our ability to start addressing these questions. And thanks to really the molecular sequencing revolution, we now know that plants interact with an immense diversity of microbial life. A large part of that comes from their soil environments. And these plant-microbe interactions could and do affect plant performance in ways that might ultimately shape our answers to this fundamental plant ecology question. So that's a lot of what I'll be talking about today. And we can start to think about capturing these plant-microbe interactions and their effects in this capacity by thinking about different functional roles that they play in the environment. So if you think about any sort of tree, maybe one outside, at any given point it might be interacting with pathogens that would have negative interactions and ultimately reduce plant fitness in certain ways. We know these types of negative interactions are important for structuring plant community diversity, like in the tropics with Jansen-Connell effects and negative density dependence. Plants are also more indirectly being affected by saprotroph, microbial saprotrophs. These are organisms that slowly recycle nutrients in the environment that then make nutrients available for plants to uptake again. We know that sort of the balance between bacteria and fungi in these soil environments helps determine the rate at which this type of cycling happens, or the sort of general energy flow cycles. So these types of microbial interactions are also important. And then finally, of course, plants interact with mutualists, microbes that provide them positive growth benefits. One type of mutualism that I'll be talking a lot about today are mycorrhizal fungi. These form really widespread symbioses between plants and fungi. 80% of plant species engage in some type of mycorrhizal symbiosis. 99% of tree species on Earth engage in some type of mycorrhizal symbiosis. These symbioses are defined by reciprocal resource exchange. So the fungi go out into the soil environment through hyphal networks. They mine nutrients from the environment in different ways, and then they send those back to the root system and trade nutrients for carbon resources that the plants are photosynthesizing. So it is this symbiotic exchange of resources. And what's interesting about mycorrhizae is they sort of hit all three of these different types of functional roles that microbes can play in the environment. They can obviously provide positive growth benefits to plants through this ability to exchange resources. They kind of act as decomposers, so they influence carbon and nitrogen cycling based on how they acquire nutrients from the environment and how they affect plant growth. And then depending on the certain dynamics of the symbioses, they can incur net growth costs to plants. So they can be more in this realm of pathogenic or negative interactions. And it's these types of functional roles that influence plant performance. And although we have a pretty good idea that these can play important roles at more local scales or individualistic scales, what I want to do is try and think about these types of microbe interactions and their effect on broader patterns of plant species distributions and potentially terrestrial ecosystem structure more generally. So I'm going to tackle this question trying to link microbial communities to the distribution and structure of plant geographic ranges. This is a huge question. There are lots of different ways you could begin to address it. I'm going to walk you through 3 examples of ways I've begun to kind of chip away at it. And then at the end, I'll present some future ideas that I have based on this work about where I think the next steps in trying to address this question are. So the first section, I'm going to be focusing on mycorrhizae, and I'm going to be looking at their effects on plant niche dimensions. So this is sort of a granular scale. How do these symbioses operate to maybe affect the mechanisms that influence plant geographic distributions through a niche-based framework? I'm then going to look at some of the patterns that these mechanisms might create at more of a global and continental scale, looking at differences in spatial variation in mycorrhizal symbioses across plant climatic ranges. And then finally, in the last section, I'm going to put these sort of processes and patterns together to think about plant-soil interactions more generally and the potential for plants to move upwards in elevation as climate change is happening in mountain systems, and how plant-soil interactions might fine-tune our expectations of exactly how plants might be able to do that. So let's jump into this first section, thinking about mycorrhizae effects on plant niche dimensions. So thinking about microbial functional roles and their potential impacts on plant geographic ranges, we can use the sort of Hutchinsonian niche framework to think about that. And the classic way that this framework is proposed is that the fundamental niche represents a set of abiotic conditions that a plant can occur in the absence of interactions with other species, so in this case would be pathogens. And then the realized niche, loosely approximating their geographic distribution because of these negative interactions, is shrunk compared to that fundamental niche. This has been sort of the classic view in ecology about how niche dynamics relate to plant species ranges, but more recently, folks have been considering positive effects of mutualisms, for example, and how they might ultimately expand plant species ranges or expand their realized niches beyond the fundamental niche limits as these mutualisms provide enhanced resource uptake or increased stress tolerance in certain host species. And so even though these types of positive interactions have begun to be incorporated into our understanding of plant niches, there still remain relatively few quantitative empirical tests of whether positive interactions actually expand plant niches. And I want to use mycorrhizal symbioses to kind of test this in a more quantitative framework. There are different types of mycorrhizal symbionts, which I'll talk a little bit more about in a second. The type I'm going to focus on right now are ectomycorrhizal or EM fungi. These types of interactions are defined by fungi that physically encase the outside of plant roots, and they kind of squeeze their way in between root cells to help trade resources there. Some of our work has suggested that 60% of tree stems on Earth engage in some type of EM symbiotic interaction, so they're incredibly widespread. And they fall under this category of kind of a classic mutualist framework in that when you inoculate plants with EM fungi, they tend to grow better in this sort of plus-minus control type experiment. So I'm going to use them to, again, try and test this niche expansion potential that mutualist interactions could accomplish. I used pine seedlings from Point Reyes National Seashore, so just north of the Bay Area. Pinus muricata seedlings. It's a coastal pine that has kind of a wonky range, but it is a good experimental tree species to use here because it does form lots of different types of EM symbioses, and we collected fungal spores from 2 of the most common symbionts, Telephora terrestris and Suillus pungens. These 2 fungi not only have really distinct morphologies above ground, but even under the microscope they create different morphologies, and there's some evidence that they are also functionally different as well. So even though trees can interact with dozens or hundreds of different EM species, we picked these two because they're some of the most common. They're functionally and kind of morphologically distinct to use this kind of test case scenario. So with this material, I created a 7x7 factorial fertilization experiment with mineral nitrogen and mineral phosphorus. So that included 49 unique treatments of fertilizer in a growth room environment. We measured plant biomass responses across this sort of two-dimensional resource space, which was replicated across 3 different fungal treatments. So a control treatment where no spores were added. So this control is, again, going to approximate that fundamental niche, so the absence of those plant mycorrhizal interactions in this case. We had a Telefera treatment and then a Pseuilus and Telefera treatment, so the combo of both of them. I did have a Pseuilus only treatment. Telefera is kind of a weedy fungus, so it got into some of the pots. So these are the 3 treatments I can reliably sort of test differences between, but I'm happy to chat about that more later. So the results I'm going to show you are 3-dimensional response surfaces, okay? So nitrogen and phosphorus are along the X and Y, and then this third axis is the total plant biomass response, so both above and below ground biomass components here. This bottom resource gradient really represents a ratio from high N to P ratios to low N to P ratios, and then the back corner of these response surfaces are where nitrogen and phosphorus were supplied in greatest amounts, okay? So just to kind of orient you to those graphs. And then I used a convex hull approach to estimate the volume of those response surfaces. And the control, again, reflecting that fundamental niche case where plants lack those positive or potentially positive interactions. And then we expect that with the addition of these traditionally characterized mutualists, they should expand the convex hull volume where plant biomass responses should be larger across a broader range of these resource gradients, okay? So here's what that looks like. So the control, I guess I can walk around a little. The control, you can see again representing that fundamental niche aspect. More yellow colors indicates greater plant biomass growth. And what this shows in the control case is that when you fertilize plants more, they grow more. If you have houseplants, that should not be surprising, right? But we wanted to know how these fungal treatments affected that. With the addition of Telefera, you can see a kind of a larger plant growth response at the top and maybe a little difference in the shoulder of this response surface over there. The total volume was 21% greater, so consistent with that niche expansion effect that we expected. The combination of both of these fungi reduced the total volume of these response surfaces, so more consistent with actual niche contraction. But we not only wanted to test the total effects of how volumes differed between these treatments, we also wanted to know exactly where they differed across this resource space. And given this design, we were able to do that by essentially comparing the difference in the control response surface to these other treatments. So these graphs, the more red color indicates a more net growth benefit of interacting with these mycorrhizal fungi, and the blue color indicates that net growth response, net growth cost. And so here what you can notice, there's 2 things I want to point out. There's obviously a great benefit of growing with Telefera versus the control treatment where nitrogen and phosphorus were in highest ratio. So there was the greatest amount of nitrogen but the lowest level of phosphorus is where Telefera improved plant growth more so than anywhere else across this resource space. And then the second thing I want to point out is that under more equally balanced fertilizers, the plant growth response was negative, so they incurred a net growth cost. So this is a little bit of a surprising result because it suggests that Telefera is this phosphorus specialist, this sort of functional specialization in this one group of mycorrhizal fungi, which are actually a pretty common group of these mutualists across the globe. So how does this compare to the dual treatment? So again, 2 things. You lose this spike in difference in plant growth response, and instead you see it kind of appear on the opposite end where nitrogen and phosphorus ratios are lowest. And you see kind of a deeper blue valley here. So interacting with both of these fungi incurred a greater net cost on plants in more areas across this resource gradient, which again, this makes sense for why you would have a negative 7% decrease in this sort of niche volume. So the start of this, we expected that these mutualistic interactions might expand plant niches across this nitrogen to phosphorus ratio gradient. One of the first things we found was that this Telefera treatment in certain cases were consistent with this idea that these mutualisms can expand plant niches based on these convex holes. And based on this design, it did indicate this sort of surprising result that one of the functional roles that this type of mutualist plays in the environment is helping plants acquire phosphorus, which could help explain their sort of broad distribution and why they can interact with lots of different tree species as well. The last two things that came out of this experiment were that at sort of moderate ratios of fertilizer, the effect of mycorrhizae really incurred net growth costs, so they acted more like pathogens, which was somewhat surprising. And then finally, when you compare the effects of both of these types of fungal treatments, that you really need to consider both the identity and the sort of environmental conditions of the symbioses to understand how they ultimately affect plant growth and might affect our interpretations along these niche dimensions, whether they expand them, whether they contract them, so that really the details of these symbioses matter. So that was the sort of first result, looking at these mechanisms of how plants interact with a certain group of microbes, how they might affect geographic ranges by influencing their niche dimensions. And now I want to focus more on the mycorrhizal communities and how they interact with tree hosts across climatic gradients, so more of the patterns that might result from these types of interactions. And to do that, I want to introduce two of the most common and broadest types of mycorrhizae that occur in nature. I've already introduced ectomycorrhizal fungi. So again, these are the fungi that form these physical structures with the outside of plant roots. Arbuscular mycorrhizae actually penetrate root cell walls and create these little tree-like structures where the resource exchange happens. And they both still send hyphae out into the environment to acquire nutrients, and they are both engaged in these symbioses. There's another interesting aspect to these, is that the trees that associate with ectomycorrhizal fungi in general have low litter quality or poor litter quality. And the trees that associate with AM fungi typically have higher litter quality, high or lowest C to N ratios. These symbioses help plants acquire nutrients in this decomposition fashion, where trees that have poor litter quality rely more on their EM symbionts to gain access to those nutrient pools, because these EM fungi, as they evolved from saprotrophic ancestors, retained much more enzymatic capabilities to degrade organic matter, things like hydrolytic and oxidative enzymes to break down those cell wall components. And so EM fungi or EM trees are able to gain more nutrients from organic matter pools thanks to the activity of their symbionts. Whereas arbuscular mycorrhizal fungi, trees that have higher litter quality, that litter decomposes more readily thanks to the activities of free-living, non-symbiotic microbes. And so they need to rely less on AM fungi to gain access to those nutrient pools. And this corresponds with AM fungi that represent the really ancestral form of this symbiosis having less enzymatic capabilities to do so. So as good plant ecologists, people started wondering, where do they grow and why do they grow there? One of the first people to ask and try and answer this question was David Reed back in 1991. And he basically drew a hand-drawn map of where different AM and EM trees occurred. And what he found was that in lower latitude, more tropical rainforests were dominated by AM trees, and more higher latitude boreal forests were dominated by trees forming ectomycorrhizal interactions. He also suggested that this climatic gradient between the equator to higher boreal forests, going from warm to wet to cool to dry, created positive feedbacks on these types of symbioses and helped explain the dominance of these different mycorrhizal states. So warm and wet conditions favor faster decomposition, especially by more free-living microbes, which would help AM trees persist in those environments because they rely less on their symbioses to gain access to nutrients. Where cold and dry climates slow decomposition, so organic matter hangs around longer in the environment, and to gain nutrients from their soils, EM trees would need to rely more on their symbionts to gain access to those resources from their greater enzymatic capabilities. So while this map is fine, it's drawn with paper and pen, I think it's fine. I love looking at maps. I think this map is fine. But the positioning of where EM and AM trees occur in nature is actually incredibly important. And that's because these differences in decomposition or nutritional mutualisms have really big effects on where carbon is, and importantly, whether it stays below ground or not. So more precisely identifying where these types of nutritional mutualisms occur on the landscape has major implications for things like global carbon cycle, whether forests are going to become carbon sources or carbon sinks under future climate conditions. And so to answer this sort of more quantitative question of whether we could identify precisely where EM versus AM trees were, I was part of a large collaboration using this giant database, the Global Forest Biodiversity Initiative database, that has over 1 million forest inventory plots where people went out, identified trees. So they physically touched 31 million different trees in this data set, so a lot of blood, sweat, and tears went into this data. With this data set covers over 28,000 tree species, and based on this data, using the best phylogenetically available data we had, we made ectomycorrhizal versus arbuscular mycorrhizal assignments to these tree species. And then using this database, we essentially asked, where do they grow? So here, this figure shows ectomycorrhizal trees on top, arbuscular mycorrhizal trees on the bottom. More warm colors indicate a greater proportion of tree basal area of these two different mycorrhizal types. So for EM trees, what you see are warmer colors in boreal forests, so a greater dominance in the boreal region, and then a gradual decline as you move south towards the equator. So again, this transition. And then for AM trees, you see essentially the opposite. You see a greater dominance in these warm, wet tropical rainforests, and then the transition to more EM forests as you move northward in latitude. So we did confirm the results from that hand-drawn map, which was good. Not too surprising, but it was good. And we noticed this global transition in mycorrhizal states. We also found that it was related to this link between climate and decomposition. So this shows temperature on the X and precipitation on the Y. The different curved lines here represent decomposition coefficients, which relate to the rate of decomposition. As you move left to right in these lines, you are increasing the rate of decomposition. And then the different colors represent proportion of EM basal area. So places that are cold and dry have greater EM tree hosts found there. And then as you move across these decomposition coefficients, you see the transition away from EM, and in this case, towards more AM dominated forests. And these decomposition coefficients were were the best predictors in the model of predicting this transition in forest mycorrhizal states. So this was a very fun collaboration to be a part of, and it was nice to sort of merge this with some historical data and think about this global transition in mycorrhizal states in the world's forests. But there's two sort of major caveats in this data set. First, we assumed that all EM trees associate with EM fungi, that there's no variance in certain trees, even if they can form ectomycorrhizal symbioses, whether they do that in different rates or not. And then secondly, there's no mycorrhizal community variation in this data set at all, which we know vary widely. And so I want to bring in more of those two components in this next section, where I've been surveying mycorrhizal communities and populace across the United States. Populace is a cottonwood trees, so Fremont cottonwoods grow around here. You probably are familiar with quaking aspen. Populus deltoides is an eastern cottonwood in blue. Populus angustifolia is a narrow leaf cottonwood in high elevation Rocky Mountain systems. And then Populus trichocarpa is a northwest cottonwood. These are incredibly important for riparian biodiversity. They grow along river systems. And they're also dual mycorrhizal. They're one of the odd cases of trees that can associate both with arbuscular and ectomycorrhizal fungi. I'm going to be focusing just on EM fungi here. We have some AM data sets, but they're not quite ready yet. So using this data, I wanted to build on that global data set of looking at sort of variation in the amount of EM symbioses that these species might have, and then also looking at the community variation of EM communities here. So over the last year or two, I've been collecting roots from almost 100 sites across the United States. This was a lot of time in a rental car by myself, so it was, I got to know myself pretty well driving around the country. Which is a story for a different time. But you can see the nice thing about these populus ranges is that they cover broad climatic gradients. And so I was in outside of Tucson in Arizona, and then a month later I was in the Mississippi, where these cottonwoods are literally flooded out by the Mississippi River. So huge, huge climatic gradients. So they're a good species to kind of study this in. From these roots, we scored them for root colonization, so levels of EM symbioses, and then we also did some sequencing, which I'll talk about. So one of the first things we noticed, so this is ectomycorrhizal colonization as percent root length on the Y, and then the different species on the X. These species vary pretty dramatically in the level of EM colonization that they had, which in this case represents, we're thinking sort of their reliance on these types of EM symbioses. This pattern matches that global latitude trend. So Populus tremuloides, quaking aspen in yellow, has a more northern distribution than Populus deltoides, which has less EM colonization. So this was nice sort of confirmatory data to show that even when we look within a single tree genus, these sort of latitudinal trends in maybe reliance on EM symbionts to gain nutrients from the environment still persists. But I wanted to know, how does the community vary as well, and how does it relate to their sort of climatic ranges in addition? So from these root samples, we sequenced them with ITS markers on standard aluminum iSeq runs. I used a Data2 workflow to process the bioinformatics side of this, and then at the end, I used Fungal to make functional assignments on the fungal communities that came out of these root samples. So we found lots of EM fungi from many different types of genera. Sampling across the United States, you might not be surprised to find a lot of EM fungi. So that was good that they do associate with lots of different types of EM partners. And I wanted to know how that variation was structured across species. So I used principal component analysis to measure that variation. So points in this graph that are closer together represent samples that share more taxa with one another, so more similar communities than points that are further apart. And what you can see is that not only do species differ in their community composition, but there's a lot of variation within species as well. And I got really curious about this because I wanted to know whether this within species variation or this dispersion in community variance within species related to their differences in geographic ranges and potentially their sort of climatic ranges. So to answer that, I went to a different database this time, the Global Biodiversity Information Facility database, that had over 9,000 georeferenced occurrence records for these 5 species. I plotted them, and then from those georeferences, I extracted 16 climatic variables, so temperature and moisture related factors. I analyzed them in a similar way as the mycorrhizal community, as I showed you previously. And when you calculate the convex area around the climatic habitats and the fungal habitats, you get sort of what I'm calling climate breadth, so the different types of climatic habitats that individuals within a species can occur in. Whereas for the mycorrhizal community, this really represents EM fungal partner breadth, so the different types of EM communities that you associate with. And then I essentially asked whether these two things were related with one another. So here we have ectomycorrhizal partner breadth and the climatic niche breadth on the Y. And then I have included a taxa plot here from the EM most common genera found, so you can kind of use that as reference. And in general, there is this positive relationship where trees that have a greater, that associate with many different types of EM communities also have some of the largest climatic niche breadth. I've also included range size here, so you can see it's not really directly correlated with range size, but really more the different types of habitats they grow in, climatic habitats. And you can see that they do, in fact, associate with lots of different types of EM. So Populus tremuloides and Trichocarpa are the bottom 2 rows here. So lots of different colors, lots of different EM partners. And you can contrast that with deltoides and angustifolia here. So also different range sizes, but they occur in relatively the same climatic habitats, and they interact with largely the same types of EM across their range. I want to be clear here that I'm not suggesting that this sort of diversity in partner breadth is causing this line. I think it's an interesting pattern. But it could very well be possible that trees that can occur in lots of different climates also encounter lots of different types of EM fungi to form associations with. So in reality, because it's ecology, it probably lies somewhere in between those answers. And I'll show some future work that I want to try and tease apart this pattern a little bit more. But it is an interesting pattern. And sort of whether we look at the global scale and these latitudinal transitions in forest states, or within a single tree genus and look at the rate of ectomycorrhizal colonization, or even start to consider the fact that identity is important. Remember from the first part of this, I showed identity was important. Whether we start to consider how identity might play into it of these mycorrhizal partners, they all indicate that this spatial variation in mycorrhizal communities is important, at least for helping us understand how plant species ranges are related to their climatic distributions. Okay, so I've talked a little bit about mechanisms. I've talked a little bit about the patterns that might occur from those mechanisms, and now I kind of want to put together the spatial variation aspect with the functional role that these microbes play in a climate change context, and think about plant-soil interactions, plant-microbe interactions with soils, in soils, across elevation ranges and simulated elevational range shifts. So if this talk started with thinking about where plants grow, why they grow there. This question viewed through a climate change lens is where will plants grow and what happens when or if they get there. And that's because plants, as their ranges become decoupled from their climatic niches, in certain places around the world are being forced to shift their distributions to track that climatic shift. And in mountain systems, that means they're gradually tracking upwards in elevation to stay within those cooler climatic niches that they've evolved in. We know that they can do this. We have great 100-year photograph differences of tree lines advancing upslope across the world, so we know it's possible. But one thing that hasn't really been taken into account as we consider that a 1 degree Celsius increase in temperature should correspond to 167 meters increase in tree range elevations. It's sort of a back of the envelope calculation, but they should move about 150 meters upwards to track their climatic shift. One thing that hasn't really been considered are their interactions with those soil environments when they get there. Those soils are conditioned by other tree species. They might be bare soil if they're expanding beyond tree lines. And really the point of this project was to think about that spatial variation in microbial communities and how it might fine-tune our expectations that plants might shift their ranges upslope. So to do that, I identified 7 populations of this Populus angustifolia species, so this Rocky Mountain cottonwood species. They occur in some of the most beautiful field sites I've ever been in, which is great. But they're also projected to increase by about 2 to 4 degrees Celsius. Lots of populations in the Rocky Mountains are expected to increase in warming. So these sites were good to test the potential range shift responses of these species and how spatial variation in soil microbes could affect their ability to do that. Across these elevation transects that I identified within each of the populations, I sampled soils directly beneath trees, so intentionally trying to collect the soil microbial communities that trees are associating with or interacting with. And then I again performed some DNA metabarcoding on these sequences to characterize the communities, and here I'll show both bacteria and fungi. One of the first questions in this project was, do microbes vary with elevation? Because if they don't, if the communities or the diversity of communities doesn't vary, then why would we expect plants to respond differently as they might move upslope? So here are showing the bacterial and fungal diversity responses to elevation, so elevation on the X and just community richness on the Y. You can see that bacterial diversity generally declines and that fungal diversity kind of peaks at low and high elevations. These are just richness estimates. We have lots of data to show that the composition of these communities also changes across elevation, not super surprising. But this was good to confirm that, yes, there is spatial variation in these communities. So now we need to understand what the sort of functional consequences of this spatial variation is. To do that, I collected plant cuttings from the field as well as gathered the soil samples that I had, and I inoculated them with either live or sterile field soil inoculum. These were gamma irradiated sterile treatments to kind of knock down or reduce as much soil biotic information or diversity was in those soils. And then I did this across all of those 7 populations across different sites along their elevation ranges, which was replicated at the genotype level. So it was a lot of plants. This was a lot of work from a lot of undergrad student researchers as well, helped me with this. And I measured the sort of range shift effect in this soil effect as the difference in plant growth in their home elevation soil versus higher elevation soil. Again, these are soil, I'm talking about microbial inoculum, so not the bulk soil, just a small teaspoon of the field soil to try and capture that microbial diversity effect. So these soil effects could be positive or negative. In this case, the way I've set it up, positive would be more inhibitory to range shifts. So if you grew better with your home microbial environment than higher elevation microbes, you could tend to maybe stay home, and that would inhibit range shifts. You could also have microbes that promote range shifts. If plants grow better with higher elevation microbes, that could facilitate the expansion of these species relative to their home soils. So So here you're seeing the soil effects on the Y, the different plant origins. I did this across different elevation stages. So low elevation plants were moved to mid elevations, mid elevation plants were moved to highest elevations, and then the highest elevation plants that we observed along these elevation transects were actually moved beyond, sort of off the front, into new soil environments where the species doesn't currently occur. So those are the different plant origins of those range shift effects, and then the different live and sterile inoculums. Again, that dashed line inhibit shifts or promote shifts. So what we see is that the direction of these effects of soils on range shifts changes depending on this gradient, this elevational gradient. So at low elevations, plants tended to grow more with their home elevation soil community. So that would suggest that range shifts at these low elevations would be inhibited by these mid-elevation soil communities. At mid-elevations, plants didn't seem to grow better or worse, depend in any of the microbial contexts. But at highest elevations, there was this dramatic negative soil effect. So plants grew much better when they were moved off the front of their current species distribution. And by that, simulating that future biotic interaction if those range edge, highest elevation plants interacted with those potential future microbial environments. I don't have time to talk about it today, but we didn't only measure plant growth responses in this experiment, we also tracked the timing of spring bud flush in this species, so getting into the greenhouse every other day, marking whether these plants were awake or still asleep through the winter. And the data that I've been working up suggests that the trend from early to late, or this phenological variation, is in part controlled by the types of microbial communities that they interact with, such that these low elevation or warm adapted microbial communities help control the early bud flush in this species, and higher elevation communities can actually reproducibly delay phenology as species might track upslope. So kind of an interesting wrinkle to this whole thing. Okay, so I've talked about sort of mechanisms that plant mycorrhizal interactions could influence broader geographic patterns of plant ranges based on this sort of niche framework and potential range or niche expansions. I've talked about, I meant to go through it like this. Sorry, and then I also found this sort of context-dependent effect where both the identity and sort of the surrounding environmental conditions influence the outcome of these interactions in ways that might not be intuitive. I've talked about how these symbioses change across plant climatic gradients, and identified that this spatial variation is important for potentially structuring where these species occur and how they interact with their below ground environments. And then I kind of put this together in the last slide, linking the spatial variation aspect to the functional effect of these interactions in a climate change context, and showed that when you expand beyond mycorrhizae and consider the sort of broader soil community, these effects on potential range shifts change as you move from low to high elevations, which could again affect our ability to predict where this species might occur under future climatic conditions. So now I want to show you some potential ideas that I have that are kind of directly bred from these lines of research. And one of the main topics is this idea relating niche dynamics to plant ranges across climate gradients. So I talked about how the relationship between the fundamental and realized niche in terms of these plant microbe interactions, how they might, in certain areas of a host range, might actually be expanding plant environmental tolerances or their resource uptake strategies. And I want to use this framework to basically link the first two sections of the talk, linking that spatial variation aspect to more of the functional mechanisms of how these interactions are occurring, to potentially define where range limits occur, or just broader range dynamics of certain species. So one way you could do that is to begin incorporating greater data on where different mutualists occur across climatic gradients. And so this would be measuring spatial variation in mycorrhizal communities, and then pairing that with more functional tests on how those community changes might actually influence plant performance across these climatic niche dimensions in ways that relate to their geographic ranges. One potential site for these types of questions would be the Sierra Nevadas, and potentially building off some of the UC natural reserves that are strategically placed in parts of the Sierras to create elevation gradients, so to define sort of plant host ranges in a experimental replicate kind of way across these climate gradients, and to track differences in mycorrhizal distributions and then relate that to plant performance. And I would be interested in using the UC Natural Reserve, not only because they have long-term climate monitoring, but they also, I saw, have some possibility for graduate student and undergraduate research funding. And just really getting more people involved in this type of really interdisciplinary project is important. You could imagine that people with more of a microbial ecology focus could be plugged in as part of this project to understand the drivers of differences in below ground organism dispersal, which is, you know, not a hard thing, or not a tricky thing to, not an easy thing to understand. So there's possibility for more of a microbial ecology role for students to identify project questions related to that. And then of course there's a role for plant ecology students, or forest ecologists even, thinking about how different populations of a forest tree might respond to different changes in climatic pressures based on this spatial structure and functional dependence on their mutualisms. Now, one small data set I have to kind of point me in this direction, again, comes from the populus data, where I found that ectomycorrhizal diversity here on the Y, so just the richness of EM communities, actually generally declines as you move away from the projected species range centers. And so this distance to range center along the X also corresponds to their distance away from their climatic niche center. So this tells me that there's something interesting happening as you move towards species ranges that structures these symbioses differently, whether the environmental conditions are harsh at species range edges and might not allow plants to fully accommodate a more diverse range of EM mutualists, or whether these mutualists, the low diversity of mutualists actually helps set these species range limits is not quite clear from this correlation. But this type of project would help me kind of unpack these types of trends of linking the spatial variation in soil communities to host ranges in this niche framework based on their performance and how they are influenced by the diversity of their microbial symbionts. So the second sort of major research direction I want to focus on kind of comes from that the last section of the talk, where I showed that the effect of soil microbial communities on plant range shifts changes with elevation. And when I first found that pattern, I was really excited because it was interesting and I could tell kind of an interesting story about that data set. But the more I've thought about it, the more I've realized how tricky it will be to try and incorporate soil microbial, the spatial variation in soil microbes into forecasting plant range shifts across the landscape. Because I've shown that they are important in these kind of elevational range shift contexts, but really I want to try and build them in sort of a bigger modeling effort so that we can incorporate this sort of spatial variation to fine-tune our predictions or forecasts of tree range shifts. One way I've thought about potentially beginning to bridge this gap is to use differences in soil feedback effects that are beginning to become apparent for EM versus AM tree hosts. So for ectomycorrhizal trees, they tend to grow better in conspecific soils, so soils that have been conditioned by the same tree species as them. And AM trees tend to grow better in soils that have been conditioned by other types of tree species. So there's a kind of a number of different research angles that are generally coming to this consensus. It's not a hard and fast rule, but there are some general patterns that suggest this. So I want to figure out if there's a way we can build on these feedback differences in different tree species by varying the dissimilarity in microbial communities between their current and future ranges in sort of a greenhouse plant soil experiment design, measuring plant growth response. I'm not going to worry about that alert right now. Measuring plant growth response across this manipulated soil dissimilarity gradient. And based on these soil feedback patterns, we would expect as you increase the dissimilarity in soil microbial communities, EM trees would grow worse because they're moving away from your more conspecific soils, and AM trees would grow better, so it might indicate this high versus low range shift potential. There's even some paleoecology data from the pollen record that suggests these differences in mycorrhizal types helped explain the expansion of plant ranges following the last glacial maximum, so I'm kind of leaning on both contemporary and historical evidence that these interactions are important for plant ranges. And then, based on the results of that type of experiment, we could use that global data set that that I was mentioning, and a freely available raster layer of these forecasts of different EM versus AM tree species compositions or tree densities across the world. And you could even consider test driving it in a California-specific example. And I would need to double check this, but I think there could be some cool synergies between using the Cal environmental DNA data set as well with strategically identifying some experiments to be used here to build some collaborations and use this data set to help basically create some of the first spatially explicit forecasts of plant range shift distributions that actually incorporate something about spatial variation in microbial communities, which I think would be fantastic. Okay, so I'm going to end where I began with this broad question of where do plants grow and why do they grow there. And through the couple research directions I've showed and future research directions, hopefully I've convinced you that thinking about the diversity of microbes below ground and how they influence plant growth in important and unique ways will be important for how we ask and answer this question in the future. And really, I think that the next phase of our ability to ask and answer this question in a fundamental plant ecology way will be to bring in more microbial ecology. And we should not be surprised if we expect to find some surprising answers as we develop this question further. So I'm happy to take any questions. Thank you all for your attention very much.
Okay, thank you, Michael. We have plenty of time for questions.
Yeah.
Yeah, so in the last part, with the elevational gradients, under climate change, the trees get to move, the microbes don't.
Yeah, which is not going to be the case. So I had to make some strategic decisions as a grad student setting up this project. And I focused on moving plants upslope, or simulating plant movements upslope, and not simulating microbial movements upslope.
Is there a logical justification for that?
No, it was it was a greenhouse space decision and a number of replicates decision. The genotype replication allowed me to ask some interesting sort of evolutionary questions about local adaptation in populations.
Yeah.
So as part of maybe the future directions area, we would be able to incorporate more of the microbial dispersal dynamics in there. And when people, I think I can think of a handful of studies that have paired that sort of transfer of the interaction upslope. They also kind of find that the outcome of the interaction changes depending on the climatic context. And so even if microbes from low elevations start to move upslope into mid elevations, that interaction kind of stays the same. But those studies don't account for the sort of mixing that might expect to happen as low elevation microbes either coalesce or compete with microbes that are already persisting in those higher elevation environments.
Is it something that one could actually, I don't know how quickly the microbial communities change. Could you do it experimentally in temperature control chambers?
Yeah.
Shift the temperature over a year or two?
Yeah, I think it's possible. I think in any type of these experiments, you're trading off some of the realism that might happen, but I think it would be possible, and I'd be interested in seeing how that might change the expectations there. Because it was a greenhouse environment. I didn't change any of the abiotic conditions. I was just focused on the biotic interaction. Yeah. Yeah.
Yeah, I thought that when we're looking at the microbial communities from like low to high elevations, did you find any like certain species that were really recurrent or like dominant that showed up in like each, or were they like really distinct?
They were distinct in the sense that the community composition changed. I haven't looked directly at, so you're asking whether there was like one common one that occurred all across the gradients. I can't think of one specific off the top of my head, but there were ones that were abundant across all samples, so I'm sure I could look into that. Yeah. Yeah.
Yes, so I'm just curious about that question you asked, because, microbes follow, especially bacterial microbes follow carbon flow. And so basically, if all your trees are moving upward, it will probably take a while to send the amount of carbon to support that population of heterotrophs to build up.
Yeah.
So is the question really of timing more than anything, rather than actually
Yeah, no, we have carbon and nitrogen data from those soils along the gradient as well, and one of the things that best correlated with the range shift effect there was how trees conditioned carbon and nitrogen differently. And so that is part of the answer, the sort of fuller answer to that. But yeah, it's definitely important. Yeah.
It looked like from the experimental setup for that final stage that you also had a really strong latitudinal gradient in the elevational gradients from which you sampled from.
Yep.
And that latitudinal gradient should also be sort of replicating the elevational gradient in terms of climate.
Yeah.
Did you test for or evaluate latitudinal effects in that experiment as well?
So I have, in the sense that I pulled out individual at the population level, so there were 7, so I can test that latitudinal trend. And latitude was incorporated in the analysis of the results as a random effect there, or as a covariate. When you look at how, at the average population, what the range shift effect was, it doesn't match the elevational trend. It's close though, so it's possible. So like lower latitude being more indicative of lower elevation, so warmer, drier climates. Yeah, I am interested in kind of measuring plant soil feedback effects across latitudinal gradients and how they compare to elevation. In this data set, it's not as clean as you would expect across elevation though, yeah. It's a good question though, and it's hard to kind of grapple with because the scale of the interactions and the latitudinal gradient are so different that yeah, it's tricky. Good question though.